This is BBIU, BioPharma Business Intelligence Unit. We provide structured insight and strategic analysis. In a world saturated with noise, surface narratives, and politically shaped data, BBIU operates at a different level of resolution. We focus on the forces beneath events: geopolitical and geoeconomic realignment, biopharma structural dynamics, liquidity stress and financial fragility, institutional drift and epistemic erosion, the architecture of power in global systems. Our work is guided by three principles: truth, structural coherence, and verifiable inference. Every model, every report, every forecast you see here is part of the analytical framework we use to brief decision makers worldwide. If you're looking for information, there are many places to go. If you're looking for structure, welcome. When assumptions replace verification. Influenza vaccine, a structural audit of DANFLU-2 and GALFLU influenza vaccine trials, executive summary. Two large real-world studies assessing high-dose influenza vaccination in adults aged 65 years or older, DANFLU Denmark, government-sponsored, and GALFLU, Spain, industry-sponsored by Sanofi, have been widely interpreted as reaching opposing conclusions regarding clinical benefit. This analysis finds that the apparent contradiction is superficial. Both studies share a more fundamental structural failure. They begin from the presumption of vaccine effectiveness, inherited from historical laboratory and regulatory claims, and restrict their inquiry to incremental comparisons between formulations. Neither study reestablishes the causal baseline required for claims of protection in a given influenza season: in-season immunogenicity, functional neutralization against circulating strains, and absolute effectiveness versus non-vaccination. As a result, endpoint choice, statistical significance, and narrative tone are shaped more by institutional incentives than by biological verification. The central scientific question, does the vaccine meaningfully work in this population in season, is never directly tested. Structural assessments sponsor context and design. Convergence DANFLU-2, sponsored by the Danish government, adopts a pragmatic registry-based design with conservative conclusions that preserve existing vaccination policy. GALFLU, sponsored by Sanofi, employs similar real-world methods but advances stronger language, including claims of superior protection, aligned with product differentiation. Despite divergent incentives, both studies converge on the same design constraint. They compare high-dose versus standard-dose vaccination only, never vaccination versus non-vaccination. This convergence is not accidental. It reflects a shared institutional reluctance to retest the hypothesis of absolute vaccine effectiveness. The missing counterfactual was feasible. Both Denmark and Galicia maintain population-wide electronic health records, with longitudinal linkage across vaccination status, hospitalizations, and mortality. A target trial emulation incorporating contemporaneous non-vaccinated individuals, propensity-weighted adjustment for baseline risk, season-specific follow-up, was technically straightforward and ethically permissible. The absence of such an analysis represents institutional avoidance, not methodological limitation. Immunogenicity assumed, not demonstrated. GALFLU states that superior protection against laboratory-confirmed influenza has been proved. However, the study reports no IgG titer measurements, neutralization assays against circulating strains, seroconversion or seroprotection rates, verification of antigenic match for the studied season. Protection is inferred from pre-existing laboratory claims, not demonstrated within the cohort itself. This reverses the correct inferential order. Immunity is presumed, protection is declared, and outcomes are retrospectively interpreted. Dose escalation without immune risk. Accounting high-dose vaccination is treated as a benign amplification of benefit. Yet higher antigen loads imply increased exposure not only to epitopes but also to excipients and innate immune activators. In older adults, characterized by immunosenescence, inflammaging, and reduced immune tolerance, annual high-intensity stimulation may carry non-trivial biological trade-offs. Neither study evaluates persistent inflammatory signatures, induction or amplification of autoantibodies, delayed immune dysregulation, nonspecific adverse outcomes plausibly linked to immune overactivation. This omission persists because it occupies a systemic blind spot protected by the public good framing of vaccines. ODP-DFP analysis, orthogonal differentiation protocol, orthogonal axis 1, biological verification. Both studies score low. Neither generates primary immunological evidence confirming neutralizing immunity in the observed population and season. Orthogonal axis 2, causal inference. Inference is constrained by the absence of a true counterfactual. Relative comparisons are performed without validating absolute effect. Orthogonal axis 3, institutional incentives. Government and industry sponsors differ in narrative objectives but converge on preserving the assumption of baseline efficacy. DFP projection, decision-facing projection. When projected forward, the current framework perpetuates marginal optimization without revalidation, limits policy adaptability across seasons, and accumulates epistemic risk as biological assumptions drift from empirical reality. The system remains internally coherent, but externally under-audited. Strategic implications. Influenza's annual antigenic drift renders historical efficacy claims non-transferable by default. Without re-establishing the full causal chain each season, incremental formulation comparisons lack decision-grade reliability. Digital health infrastructure already enables the analyses required to close this gap. Their absence reflects governance priorities, not scientific impossibility. Conclusion: DANFLU-2 and GALFLU do not represent conflicting scientific truths. They represent a shared epistemic omission. Both operate within a framework where vaccine effectiveness is treated as axiomatic rather than empirical. Incremental differences are measured, while the foundational premise remains unexamined. Until influenza vaccine studies reestablish the full causal chain, from in-season immunogenicity and functional neutralization to laboratory-confirmed infection and clinical outcomes, claims of superior protection will remain incentive-aligned assertions, not verified conclusions. This is not a failure of data access. It is a failure of epistemic discipline. Annex. Methodological and epistemic improvements toward decision-grade influenza vaccine evidence. BioPharma Business Intelligence Unit, BBIU, Technical Annex. Improvement Framework. A1. Reestablish the causal chain. Before measuring marginal effects. Problem identified. Both DANFLU-2 and GALFLU measure incremental differences without reestablishing whether the vaccine is biologically effective in the observed population and season. Improvement required. Reorder the inferential logic, in-season immunogenicity IgG titers measured post-vaccination, seroconversion and seroprotection rates, functional validation, neutralization assays against circulating seasonal strains, explicit documentation of antigenic match-mismatch, biological efficacy, laboratory-confirmed influenza incidence, PCR-based, clinical translation, hospitalization, complications, mortality. Without steps 1-2, steps 3-4 cannot support claims of protection. A2. Incorporate a non-vaccinated counterfactual using EHR infrastructure. Problem identified. Absolute vaccine effectiveness is never retested despite full digital health records. Improvement required. Implement target trial emulation using existing data. Eligibility defined at season start. 3 strategies: no vaccination, standard dose vaccination, high dose vaccination. Propensity-weighted adjustment for age, comorbidities, prior vaccination history, baseline healthcare utilization, uniform follow-up window. This design avoids placebo ethics, removes sample size constraints, allows absolute and relative effectiveness estimation. A3. Separate administrative outcomes from biological outcomes. Problem identified. Hospitalization codes are treated as proxies for biological protection. Improvement required. Explicitly distinguish biological outcomes, PCR-confirmed infection, viral load, symptom duration from administrative outcomes. Hospitalization, pneumonia, coding, cause of admission, ambiguity analyses should present biological outcomes as primary evidence, administrative outcomes as downstream translation, not the reverse. A4. Add immunological subcohorts. Each season. Problem identified. Population-level trials generate no immune-level verification. Improvement required. Predefine representative immunological subcohorts, stratified by age, sex, frailty, comorbidities. Serial sampling, baseline post-vaccination, 2 to 4 weeks. Late season. Measured endpoints, neutralizing antibodies, breadth of response, persistence of immunity. This converts registry trials from black box outcome studies into mechanistically interpretable systems. A5. Explicitly evaluate dose-risk trade-offs in older adults. Problem identified. High-dose vaccination is assumed to be risk-neutral. Improvement required. Systematically monitor and report markers of systemic inflammation, autoantibody induction or amplification, delayed immune dysregulation, nonspecific hospitalizations temporally associated with vaccination. This does not imply expected harm. It restores benefit-risk symmetry to dose escalation decisions. A6. Require sponsor-independent revalidation of baseline efficacy problem identified. Both public and industry-sponsored trials inherit manufacturer claims as axioms. Improvement required. For each influenza season, baseline efficacy must be re-estimated independently of formulation comparisons using contemporaneous data. Incremental superiority claims should be conditional on demonstrated baseline effectiveness. A7. Pre-register epistemic, not only statistical, criteria problem identified. Trials pre-register statistical endpoints but not inferential thresholds. Improvement required. Pre-specify what constitutes biological protection, what level of neutralization is considered meaningful, how discordance between immunology and outcomes will be interpreted. This prevents post-hoc narrative stabilization. A8. Integrate ODP-DFP as a decision filter orthogonal differentiation protocol, ODP. Future influenza vaccine trials should be evaluated across orthogonal axes: biological verification, causal inference integrity, population specificity, risk transparency, incentive alignment, decision-facing projection, DFP. Policy conclusions should only be drawn when orthogonal coherence is achieved, not merely statistical significance. Annex conclusion. The limitations observed in DANFLU-2 and GALFLU are not technical failures. They are the result of a system optimized for continuity rather than revalidation. Digital health infrastructure, modern causal methods, and immunological tools already exist to close these gaps. Improving influenza vaccine evidence does not require new technology. It requires restoring epistemic discipline and accepting that effectiveness must be demonstrated repeatedly, not presumed indefinitely. Annex 2. Current technical framework. How seasonal influenza vaccines are designed today. B1. Global surveillance as the starting point. Seasonal influenza vaccine design begins with global viral surveillance, coordinated primarily by the WHO Global Influenza Surveillance and Response System. Key elements: 150 national influenza centers, NX, continuous sampling of circulating influenza A and B viruses, genetic sequencing and antigenic characterization, monitoring of strain prevalence, geographic spread, emerging drift variants. This system does not predict future strains with certainty. It maps current and recent circulation. B1A. Data origin and transparency characteristics. Primary surveillance data are collected at the national level through NX designated by the WHO. These centers receive clinical samples from hospitals and sentinel sites within each country. Key characteristics. Sampling intensity and representativeness vary by country. Case ascertainment depends on national testing policies and capacity. Only a subset of detected isolates is forwarded for advanced analysis. Aggregated surveillance outputs, e.g., weekly counts, subtype distribution, are publicly available through WHO platforms such as FluNet, while viral genomic sequences are shared through repositories such as GSAID. However, raw patient-level clinical data are not publicly released. Denominators, who was tested versus not tested, are incompletely transparent. Criteria for isolate prioritization are not fully standardized across countries. B2. Antigenic characterization and drift assessment. Collected viral isolates undergo hemagglutination inhibition (HI) assays. Genetic sequencing of hemagglutinin (HA) and neuraminidase (NA) assessment of antigenic drift relative to prior strains. The goal is to identify dominant circulating lineages, significant antigenic changes that may reduce prior immunity, candidate strains likely to dominate the upcoming season. This process is probabilistic, not deterministic. B3. WHO vaccine strain selection process. Twice yearly, WHO convenes expert consultations. February, Northern Hemisphere recommendation. September, Southern Hemisphere recommendation. Based on surveillance data, WHO recommends 3 strains trivalent or 4 strains quadrivalent, typically influenza H1N1, influenza H3N2, influenza B Victoria lineage, influenza B Yamagata lineage, increasingly absent post-COVID. These are recommendations, not mandates. B3A. Determination of predominance within the strain selection process. The designation of a strain as predominant is not based on prevalence alone. It reflects a composite assessment of geographic spread and trajectory, antigenic drift, significance, epidemiological momentum, temporal feasibility for manufacturing timelines. As a result, predominance is a technical determination, not a purely statistical label. B3b, interface with manufacturing feasibility. Although pharmaceutical manufacturers do not participate in primary surveillance, industrial feasibility enters the process at this stage. Specifically, candidate strains are evaluated for growth characteristics, egg or cell-based platforms, antigenic stability during propagation, expected production yield timeline, compatibility with seasonal deployment. Strains that are biologically relevant but poorly manufacturable may be deprioritized. This influence is structural rather than directive. Manufacturers do not select strains, but production constraints bound what can realistically be advanced. B4. Translation into national and regulatory decisions. Regulatory authorities, FDA, EMA, MFDS, etc., adopt WHO recommendations with minimal modification. Key characteristics: no requirement to reprove absolute clinical efficacy each season. Emphasis on antigenic similarity, manufacturing feasibility, continuity of supply. Regulatory review focuses on strain change validation, manufacturing consistency, safety comparability, not on de novo efficacy trials. B5. Manufacturing platforms. Most seasonal influenza vaccines are produced using egg-based manufacturing, fertilized chicken eggs. Viral adaptation may introduce antigenic changes. Long production timelines. Cell-based manufacturing. Mammalian cell lines, reduced egg adaptation risk, still limited global capacity. Recombinant HA platforms, HA protein expressed in insect or other systems. Faster scalability, less widespread adoption. The platform does not alter strain selection logic, only production characteristics. B6. Antigen dose and formulation strategy. Standard dose vaccines contain a fixed amount of HA per strain. High-dose formulations increase HA concentration per strain. Rationale: counteract immunosenescence, increase antibody titers, improve seroprotection rates. Key point: dose escalation is justified by immunogenicity metrics, not by repeated seasonal efficacy trials. B7. Immunogenicity as the regulatory surrogate approval and annual updates rely on hemagglutination inhibition titers, seroconversion rates, seroprotection thresholds. These are treated as validated correlates of protection, despite seasonal variability, strain mismatch risk, population heterogeneity. Clinical outcomes are not routinely revalidated each season. B8. Deployment and policy implementation. Once approved, vaccination campaigns are launched. Pre-season coverage targets drive success metrics. Effectiveness is assessed retrospectively via observational studies. Policy emphasis is placed on uptake, logistics, risk group prioritization, not on real-time biological verification. B9. Summary of the current system logic. The current influenza vaccine system operates on the following logic. Global surveillance detects circulating strains. Expert committees select candidate strains. Manufacturing feasibility constrains translation. Regulators approve updates based on immunogenicity and continuity. Vaccines are deployed at scale. Effectiveness is inferred retrospectively. This framework prioritizes speed, scalability, supply stability over season-specific biological revalidation, individualized response assessment. B10. Institutional architecture and distributed governance. The global influenza vaccine system is governed through a multi-layered institutional architecture in which authority, funding, procurement, and execution are intentionally separated across distinct organizations. This structure is not accidental. It reflects decades of attempts to balance scientific authority, political legitimacy, financial capacity, and industrial execution at global scale. B10A. World Health Organization. The WHO functions as the normative and technical authority of the system. Its core responsibilities include defining global surveillance frameworks, e.g. GIS, convening expert consultations for strain selection, issuing technical recommendations and position papers, coordinating reference laboratories and standards. Critically, WHO does not finance vaccination programs, does not procure vaccines, and does not implement national immunization campaigns. WHO's authority is therefore epistemic and coordinative, not operational or financial. B10B. Strategic Advisory Group of Experts on Immunization, SAGE. SAGE is an independent advisory body convened by WHO. Its mandate includes issuing global policy recommendations on vaccine use, defining target populations, e.g. elderly, high-risk groups, advising on prioritization during constrained supply. Key characteristics: members are required to declare conflicts of interest, deliberations are consensus-based, recommendations are non-binding. SAGE does not design vaccines, select strains, approve products, or manage procurement. Its role is policy framing, not execution. B10C, WHO Pre-Qualification Program, PQ. The pre-qualification program serves as a technical gatekeeper for global vaccine supply. Functions include assessing manufacturing quality and consistency, reviewing safety and production standards, enabling vaccines to be purchased by UN agencies and global donors. PQ does not conduct efficacy trials, determine clinical benefit, or compare vaccine effectiveness. Its focus is manufacturing reliability, not biological performance. B10D. Gavi, the Vaccine Alliance. Gavi is a financing and market-shaping organization, legally and operationally independent from WHO. Its responsibilities include mobilizing donor funding, negotiating vaccine prices and volumes, determining which vaccines are financed for eligible countries. Governance characteristics: public-private partnership structure, participation of governments, WHO, UNICEF, World Bank, philanthropic actors, and industry representatives, non-voting. Gavi does not generate scientific recommendations, conduct surveillance, or evaluate biological efficacy. Its decisions are conditional on WHO recommendations. B10E, UNICEF. UNICEF acts as the primary procurement and logistics agent for vaccines financed by Gavi and other multilateral mechanisms. Responsibilities include issuing tenders, signing supply contracts, managing global distribution and cold chain logistics. UNICEF does not define vaccine policy, assess effectiveness, or select strains. Its mandate is execution and delivery. B10F. National regulatory authorities, national regulators, e.g. FDA, EMA, MFDS, retain sovereign authority over vaccine approval and use. In the context of seasonal influenza vaccines, approvals rely on strain change supplements, immunogenicity, and manufacturing comparability are emphasized. De novo efficacy trials are generally not required annually. Regulators typically align with WHO recommendations, international harmonization standards. B11. Responsibility distribution and structural accountability gap. Across this architecture, scientific guidance is issued by WHO and SAGE. Financing decisions are made by Gavi and national governments. Procurement and logistics are handled by UNICEF and national systems. Manufacturing execution is performed by industry. Regulatory authorization is granted by national agencies. Each institution operates within a clearly defined mandate. However, no single institution retains end-to-end accountability for reassessing baseline biological effectiveness each season, auditing the full upstream decision chain from field data to strain selection, or systematically revisiting inherited assumptions when new data capabilities emerge. Responsibility is therefore distributed, shared, but not centralized. This creates a structural condition in which resources and authority flow efficiently, while epistemic accountability remains diffuse. Annex 2 Conclusion. Seasonal influenza vaccine design is not a failure of science, but a system optimized for probabilistic control under time constraints. Its technical architecture assumes historical validity of immunogenicity surrogates, acceptability of uncertainty tolerance for seasonal variability. While surveillance data are largely transparent in aggregated form, the decision logic translating field data into vaccine strains involves technical weighting steps that are only partially visible externally. This governance model prioritizes robustness, scalability, and continuity. At the same time, it complicates comprehensive accountability, as no single actor is structurally mandated to challenge foundational assumptions once the system is in motion. Understanding this framework is essential before evaluating where, why, and how it may require modernization. Annex 3. Pharmacoeconomics and industrial structure. Seasonal influenza vaccines, market size, relative weight, manufacturing geography, and market dominance C1. Global market size of seasonal influenza vaccines. The global market for seasonal influenza vaccines represents a large, recurrent, and structurally stable revenue stream within the pharmaceutical sector. Across multiple industry analyses, current annual revenues converge within a narrow band. The seasonal influenza vaccine market is estimated at approximately USD 8 to 9 billion per year at present. Medium-term projections indicate growth toward USD 12 to 13 billion by the end of the decade, driven primarily by aging populations in high-income countries, expansion of high-dose and adjuvanted formulations, increased policy emphasis on respiratory disease prevention post-COVID. Unlike many vaccine segments, influenza demand is non-episodic. It recurs annually, independent of outbreaks, which confers exceptional predictability to manufacturers and purchasers. C2. Share of influenza within the global vaccine market. The global vaccines market as a whole is estimated in the range of USD 65 to 85 billion annually, depending on inclusion criteria, routine pediatric vaccines, adult vaccines, travel vaccines, pandemic stockpiles. Within this context, seasonal influenza vaccines account for approximately 10–13% of total global vaccine revenues. This places influenza among the largest single vaccine categories worldwide, comparable in economic weight to entire therapeutic clusters. This proportion is notable because influenza vaccines do not rely on outbreak-driven demand, are supported by standing public health recommendations, and benefit from entrenched procurement mechanisms. From a pharmacoeconomic perspective, influenza vaccines function as a core revenue anchor within vaccine portfolios rather than as a marginal product. C3. Manufacturing geography and capacity. Seasonal influenza vaccine manufacturing is highly centralized, capital-intensive, and geographically concentrated. Key structural characteristics include: global production is carried out by approximately 30 manufacturers, but more than 85% of worldwide supply is produced by fewer than 7 manufacturers. Manufacturing capacity is unevenly distributed. Europe and North America host a large proportion of legacy egg-based and cell-based facilities. Asia-Pacific, Japan, China, South Korea, Australia, India represents a growing share of both bulk production and fill-finish operations. Latin America maintains regional manufacturing and fill-finish capacity, often linked to national immunization programs. Global seasonal production capacity is estimated at 1.5 billion doses per year, a figure that has remained relatively stable since before the COVID-19 pandemic. This capacity constraint is critical. Influenza vaccines must be designed, manufactured, released, and distributed within a fixed annual window. Industrial feasibility is therefore inseparable from strain selection. C4. Dominant market participants. The influenza vaccine market operates as a supply-side oligopoly. A small number of multinational manufacturers dominate global production, distribution, and regulatory engagement. These include Sanofi, CSL, Seqirus, GSK, AstraZeneca, primarily live attenuated platforms. Alongside these, several state-affiliated or regional manufacturers supply domestic or regional markets, often under technology transfer or licensing arrangements. While these regional producers are important for local supply security, global market dynamics are set by the dominant manufacturers, who control the majority of bulk antigen production, platform know-how, egg-based, cell-based, recombinant, and global regulatory interaction. C5. Pricing structure and economic segmentation. Seasonal influenza vaccines are not economically homogeneous. The market is segmented into standard dose inactivated vaccines, high-dose formulations, adjuvanted vaccines, and live attenuated vaccines. High-dose and adjuvanted products command price premiums, particularly in older adult populations, justified primarily by higher antigen content, enhanced immunogenicity metrics, and policy-driven recommendations for elderly cohorts. Pricing is largely determined through public procurement and tenders, national immunization program negotiations, and reimbursement frameworks in high-income countries. This structure reinforces predictable margins and reduces exposure to classic market competition. C6. Structural implications for the upstream decision chain. Given the size, concentration, and predictability of the influenza vaccine market, strain selection decisions carry non-trivial economic consequences. Manufacturing feasibility becomes a binding constraint rather than a secondary consideration, and continuity of supply is prioritized over experimental variability. This does not imply improper influence, but it does establish a structural dependency between global surveillance outputs, strain selection feasibility, and industrial execution. In pharmacoeconomic terms, seasonal influenza vaccines represent a systemically important product class, too large to fail operationally and too recurrent to tolerate disruption. Annex 3. Conclusion. Seasonal influenza vaccines occupy a unique position in global pharmacoeconomics, economically significant, structurally concentrated, and operationally time-bound. If your organization requires structural clarity, BBIU is available for private briefings and strategic analysis. To initiate contact, visit BioPharma Business Intelligence Unit.com and reach out through the Institutional Access Channel.